Okay, so hello and welcome back to Excelsior Academy. So this topic is really important because a few questions have come from this topic in the previous question papers. So let's talk about the REC BCD pathway, and as I mentioned, it is in the prokaryotes. So in the previous lecture, we have discussed like how in the case of eukaryotes, the recombination is happening. Okay, so if you remember in the last lecture, we have discussed about the holiday junctions. Okay, so if this is clear, then only this will be clear to you. Okay, if the concept of holiday junction is clear to you, then only the reg BCD pathway will be clear. So here the different proteins were there. In the case of eukaryotes, homologous recombination was happening, and it was giving you two types of product. So Chandni, can you tell me what are those two types of product which you get in holiday junctions? If there is a vertical break or if there is a horizontal. So in both the scenario, what kind of products you are getting? Remember anyone? Remember? What about others? Akshay, Tanvi, do you remember?
Yes, ma'am.
So what is happening if you are having a vertical cut in at the
Ma'am, sorry, ma'am. Crossover recombinants are formed.
Yes. Okay. So those are also called your splice recombinants, correct? So crossover ho gaya ya splice ho gaya. So okay, good. Now whenever there is a horizontal cut, then it is your non-crossover products, or it is also known as patch recombinants. Okay, so yehi concept mein you today we'll be doing our reg BCD pathway. Okay, so we'll be going very slowly because the diagrams are complicated, and as such a good video I didn't get, otherwise I would have put that. Okay, so this is happening in the case of the bacteria. Okay, so both the rec D and okay, as the name suggests, it is rec B, C, D. So these are the three different proteins, okay? B, C, D. So here you need to remember is D and B are helicases, which are like energy dependent molecular motors. So what do you mean? Energy dependent means they are going to use ATP, okay? When you say molecular motors, what does it mean? They are, with the help of the ATP, they are going to move on to the DNA sequences. Anything which can walk on DNA. So if you remember, in the case of cell biology, there are certain cytoskeleton proteins are there. Okay, I hope you must have heard about myosin. Then there are certain something called kinesin and dynein. Have you heard of these three different types of proteins? Have you heard of these?
Yes, ma'am.
Yeah. So myosin is your muscle motor protein, correct? Which helps in the contraction and relaxation of the muscles. So this comes under your microfilament, okay, made up of actin and others. Then comes the kinesin and dynein. So they are your tubular proteins, tubular motor proteins, which is again microtubules. Okay, so they are microtubules, motor proteins, which help in the movement of the organelles from inside to outside and outside to inside. So similarly, your rec D and B are motor proteins, which with the help of ATP, they are driving. Okay, so it is important how they are going to function. Of course, even they need ATP. Any motor proteins, they are going to require ATP. Okay. So what is the function? They act like a helicase which unwinds the DNA. Okay. So rec B subunit in addition has nuclease function. This is nuclease function. Please remember. So among B, C and D, B and D has got helicase activity. Again, B has got nuclease activity. Now the question comes is based on the rec B and rec D function. Okay. Now what is the function of C? C का function है to recognize a specific sequence called chi. ऐसे symbol बनाते हैं, right? Something like this. Oh, okay, like this. Chi we say. Okay. जो chi square में हम कहते हैं ना, it's the same chi. So rec C subunit, the main function is to recognize this chi, which is actually a hotspot where the recombination happens maximum. Are you able to follow? D and B are helicases where rec C, okay, rec C is recognizing a specific sequence in DNA which is known as chi. That is crossover hotspot instigator. इसका मतलब क्या? DNA में एक ऐसा sequence रहेगा आपका around 8 nucleotides का which is like hotspot where the crossing over is going to happen. ठीक है, जहां पे आपका recombination होता है, जहां पे आपका homologous recombination will be happening. So we'll talk about this chi also in detail. So what is the takeover message over here? That you need to remember the function of rec B, C, and D. Okay, can you remember? Now this is also important that rec B has got 3-5- helicase activity. So what does it mean? The direction in which it is going to move. Okay, then D ka hai aapka 5' to 3' direction. So this slide is very, very important for you with respect to your exam. Okay, point of view. Clear? Can you guys remember this? Because based on this only, based on this information only, the upcoming diagrams, it will be clear. Okay, so should I go through once? Are you able to remember? Simple, rec BCD we say, right? So rec is standing for recombination. B, C, and D, it's for the subunits. B and D helicase activity, B in 3' to 5', D in 5' to 3' activity. B also has got the nuclease activity. What is the function of nuclease? It is going to chop off the nucleotides. ठीक है, जहां पे भी break होगा. So this rec BCD pathway is for what? Okay, that okay, sorry, I should have mentioned before like why this rec BCD pathway is there. So rec BCD pathway is there in order to repair double strand break in prokaryotes. So if you remember in the previous lecture, we have discussed about the holiday junction, we have discussed about the measles and Radding model also, and then the double stranded break model. So how in the case of prokaryotes it is happening is called the rec BCD pathway. Clear? From the previous lecture to this lecture, are you able to relate? Yes? Okay. So the rec BCD pathway is meant to repair double strand break in prokaryotes. Okay, now this is clear. So why we are discussing about the rec BCD? Now the function of rec BCD. So B is having the helicase as well as nuclease activity. C is having it recognizes the chi sequences, and D for the again 5' to 3' helicase activity. Okay. Now let's see the how it is going to proceed. So first step is what? That in the rec BCD pathway, you have got the homologous chromosome and there is a double strand break, okay, in one of the DNA molecule. So what do you see over here? When the break is like this, so it has got the something called blunt ends. So I hope everyone knows what is blunt ends, correct? The same cut will be here. It is not having the sticky ends like this. Okay, this is known as sticky ends. So overhangs aate, jisme 3', 5' overhangs aate. But here you don't have overhangs. Your 5' is over here, your 3' is over here, and 3' here and 5' here. So blunt ends it is. Okay. Now, what happens now is that when there is this double strand break is there, then in order to process this DNA, okay, the blunt is your rec BCD subunit proteins comes and binds over here. Okay, so can you see it over here? D and BC, it's together. Okay, so BC, they have made it like this over here, but they are separate subunits. So you have got the rec B, rec C subunits and rec D subunit. Correct? Okay, so they come and recognizes the where the blunt ends are present. Okay, now what is expected now? What should happen? If you see, we remember that B and D, okay, B and D has got the helicase activity and B is also having the nuclease activity. Okay, so if you see what is happening, as long as your helicase activity is happening, ठीक है, जैसे जैसे आपका helicase activity दिखा रहा है, sidewise the B, okay, it is also showing its nuclease activity. So जो भी DNA cleave हो रहा है, that is being destroyed, that is converted into the nucleotides. Okay, is this figure is clear? So because of this nuclease activity, what is happening? A gap is introduced. Okay, clear? Able to follow me?
Yes, ma'am.
Okay, so one strand is being destroyed, okay, and the other one is forming the loop. Okay, now what happens over a period of time? You can see this lower strand is getting chopped off. Okay, now tell me in which direction the BCD complex is moving. Yes, in which direction?
It's an anti-clockwise direction. No?
3 prime or 3 prime to 5 prime? In DNA, we don't talk about anti-clockwise or clockwise. We talk about 5 prime to 3 prime or 3 prime to 5 prime direction. So since you can see the BCD complex is moving in this direction, so what is the direction over here? Huh? It is clear from the figure. Akshaya, what do you think? What is the answer?
5 prime to 3 prime.
Yes, see, you can see 5 prime to 3 prime. It is moving like this. So it is 5 prime to 3 prime direction. So it means your D is activated. That is your D. If you remember, B has got your 3 dash to 5 dash helicase activity. ठीक है? इसलिए मैं कह रही हूँ याद रखना है. Now what happens? After certain sequence, when this is chopped off by the activity of B subunit, okay, the chi side has come. Clear? This much is clear. So this sequence is going to now recognize this side. Now, whose function it is to recognize the chi side? It is a function of your C. Okay, simple to remember. C is for your chi side recognition. Okay, clear. Now, so chi sites are the recombination hotspots in the genome of E. coli bacteria. This is the sequence. No need to remember. No, not at all required. Now, what is important to remember is that when reg BCD encounters a chi sequence, the function changes. Okay, from exonuclease to a helicase, producing a single strand of DNA that invade homologous double strand DNA mediated by rec A. Abhi kya tha? Sirf BCD tha. Now the function of rec A is going to come. Now how this is happening? Okay, can you see now the function of D has been stopped as it is crossing the chi side. Aapne dekha, right? Now if you see clearly over here, we see that there is a gap also produced over here. Such a big gap is there. Correct? Now, is this much is clear that it has sequence, it has started. So D ka 5 prime to 3 prime helicase activity has stopped now. This much is clear?
Yes,
Okay, itna clear hai, right? So, iska matlab kya? Now, further they are not moving. Okay? The function of C is been done, the function of B is done, the function of D is done. So further there is no movement. It means further there is no helicase or unwinding of DNA. This much is clear. Now, do you remember the strand invasion in the case of holiday junction? What is strand invasion? Do you remember? So one of the strands over here is going to invade here, having the similarity in the sequences. That is like homologous sequence. So it is going to invade the strand. Okay. Can you see this is your 3' single strand strain is there. Now this is going to invade the homologous strand. Now before that, in order to help in the strand invasion, your rec A protein is coming. Okay. Now, this is also important to remember because calculations comes for this. So one rec A protein is going to bind the 3 nucleotides. Can you remember this much? Okay. So what is happening? The moment this is free before the strand invasion, that is like in order to initiate strand invasion, rec A protein will come and it will start binding the single strand DNA like this in a helical manner. Okay. Just remember the figures as if it is going to play in the background. Okay. Just imagine like that. Now, can you see that it has bound to this DNA in a helical manner? Can you see? Yes?
Yes, ma'am.
Okay. Yeah, okay. Now, look at this figure. So we have zoomed it over here. And what do we see here is that this is this red color, sorry, this green color, big sphere are your Rec A protein. Okay. So of course, when a single monomer comes together, it is known as filament. So now what had happened is, since it has been bind like this, it is now known as filament. Now it has got a primary binding site and a secondary binding site. Now this is important, okay? So can you see this nucleotide? Which one is it is? This is your A, correct? A. So in this protein together, in the primary binding site, it is having A. Okay. Now it is going to search, okay? This Rec A filament is going to search in these two homologous sequences where you will have A T base pairing. Okay, समझ में आ रहा है? It is going to search over here where the sequence of this is going to match over here. So what will happen? This pink and the black strand will come in the complementary sequence. Are you able to follow? नहीं? सी मैं फिर से बोलती हूँ, ठीक है? थोड़ा सा ध्यान दो यहाँ पे. Can you see the direction of the sequences over here? Like that, it has been shown, right? The arrow is like this. Agree? So what does it mean? It means either it is 5 prime to 3 prime, तो नीचे वाला होगा आपका ऐसे. Okay, this is how we represent in the diagrams, like one arrow this side, another arrow this side. So these two strands are clear to you. Okay. Now this pink strand, can you see what is the direction? It is this direction is there. So obviously, when base pairing should happen, it should happen with which strand? Black or gray? Black or gray? Which direction then it should be? Tanvi, समझ में आया?
Yes, ma'am.
तो कौन से स्ट्रैंड के साथ वो बेस पर करेगा? पिंक वाला. If I remove these two, I'm breaking the hydrogen bonds between the black and the gray strands. Then this pink strand is going to base pair with which strand? Black or gray?
Black.
Yes, it is black, right? So these two are going to be in the same direction. They won't pair. So this pink one is going to replace your gray and going to base pair with black. ठीक है? तो ये बीच में यहाँ आएगा. So this is how the strand invasion will start. Now, how exactly this is happening? Okay. So can you see the Rec A has inserted itself into between these two black and gray strands? Correct? Now it will keep on scanning the sequences till it get a correct base pairing. So once it reaches a base pairing, like it is having A in its primary pocket. See, it is having A in its primary pocket, okay? This is its primary pocket. This is its secondary pocket. So secondary pocket means where you have got the A-T ka base pairing. So it is going to scan the sequences in the homologous chromosome where you are going to have A and T. Okay. Now, in order to complete the strand invasion, what Rec A protein is going to do? It is going to break like this. Okay. It is going to break the base pairing between A and T. Okay. Now, so this is what it is scanning till you are going to get a proper sequences. Can you see? It is black, pink, and gray. So they keep on breaking the bonds between the black and gray till they get a perfect match. Now, is this a perfect match? AAT आपका perfect match हो रहा है? And remember, pink has to base pair with black. So is it happening? The perfect match?
No.
No, ma'am.
No, because A and A cannot form base pair. So it is going to skip this. Okay. Don't get confused that AT hai to matlab ye hona chahiye. Aisa nahi hai. Keep in mind that pink and black has to base pair. So A and A cannot base pair. Now, again, it is keep on scanning the sequences. Like, see, let us say that these are the sequences. It keep on scanning. Next, it encounters GC. Is it going to base pair? No. A na G के साथ हो सकता है ना C के साथ होता है, right? So it is going to skip this base pairing also. Then next, now it has got this base pairing. So what do you think? If the bond is broken between these two, can this one and this one will base pair? Yes, it is going to base pair. Correct? Now onwards, the proper we can say that the strand invasion has taken place. इसका मतलब ये नहीं कि सिर्फ बीच में घुसने से हो जाता है. It needs to get stabilized, right? It needs to form a base pair with the upper strand. So this is how your strand invasion happens. This is like in detail. अभी clear है कैसे होता है? So we have taken only one example. So Rec A protein, which is like helping in one. So you can see that many Rec A proteins are there, which totally finally helps in proper binding, proper base pairing between the black and the pink strand. Clear? Is this much is clear? So it keeps on scanning till the proper base pair match has been done. Okay? See over here. You can see. So TA can base pair by breaking the bond between this A and this T. So this pink strand has come in between. Okay? Now it is clear?
Yes.
Okay? हाँ. You need to respond, otherwise मुझे पता नहीं चलेगा whether understood or not. So this is how the base pairing will be done, and finally the strand invasion is being done. Okay, so this is how it is going to look. Now I have just put this slide in order to, you can, so that you guys can compare like how exactly we have studied in holiday model. Okay, so in the holiday model also we have seen that this particular crossover junction comes and then it rotates so that finally the proteins can decide whether we can have the vertical cut or the horizontal cut. So same way over here also, the only difference is that in the case of Rec BCD pathway, the chances is having two crossover, okay, two crossover or two holiday junction is possible. So like this. So अब क्या होगा? Because of this, ये ऐसे जाके ऐसे आ जाएगा. So here you have got one holiday junction and here it will form another holiday junction. So this is similar to your holiday junction. Okay? So this much is clear. So now you know how the strand strand invasion has been done with the help of the Rec BCD. So you can see it over here. So finally, वो यहाँ पे strand open कर देगा and it will bind. Now what you need to remember is that जहाँ gap है, it is not like that you have got over here. Remember, initially the Rec B protein, which has got the nucleus activity, has created the gap, right? Remember? So because of this gap, now in order to form a two holiday junction, there is a still gap is there. That too in two strands it is there. So it is going to recruit your DNA polymerase so that it can fill the gap. And we know that this filling of the gap is done by the DNA polymerase 1. Clear? So it is going to fill the gap over here. So यहाँ पे भी वो fill करेगा and सारा यहाँ पे भी fill करेगा. Okay? And then of course we know that the next protein is ligase, which is going to come and reseal it. So yes, so यहाँ पे भी seal हो गया, sorry, and यहाँ पे भी seal हो गया. Now we don't have any double strand break, but is it now over? Do you think it is over? No, right? Because the chromosomes are tangled now. They are in their holiday junction scenario. Correct? Guys, are you able to follow? Stop me. We can go back. Okay, we can do it again. No problem. So till now, what we have seen that we have got the homologous sequences are there. Okay, And when there is a double strand break is there, then this reg BCD protein comes and binds over there. So the function of B and D is helicase activity. They keep on breaking the bond. B nuclease activity se wo chop off karta rehta hai. It removes the unwanted nucleotides, right? Then when it reaches the chi site with the help of the reg C protein, okay, then it is going to stop. So the activity of D stops, and later it is going to get bind with the reg A. Reg A protein is going to help in the strand invasion with the homologous sequences. Okay, so we have seen how the strand invasion is being done at the molecular level. Okay, now once it is being done, then you can find at least minimum 2 holiday junctions forming. Okay, and the gap is filled by the DNA polymerase 1, and ligase will come and seal the gap. Okay, seal the nick, sorry. Now the thing is that this is not yet done. So we need to open up the structure. We cannot have like this, the as a though homologous sequences tangled like this. We can't have like this. So we need a something which can separate. And for that, we have got the protein called resolvase. The name of the protein is resolvase, which is made up of these proteins. Okay, so RUV A, B, and C. जिसमें से C का मेन काम होता है to resolve. Now, what does A and B is doing? A and B is helping in recognition of the holiday junctions. Okay, so A and B, they help in recognition of your holiday junctions. Okay, so in short, they're also known as the branch migration. So if you remember in the case of holiday junction also, it's not that it will stop over here. This entire branch will shift over here, and this might shift over here. Jitna maximum possible ho sakta hai. Okay, so they are going to separate out. Now, why do you think this is important? Because as they migrate, the chances of having more recombination becomes high. Correct? तो अभी क्या होगा, यहाँ पे सिर्फ इतना शेयर होगा. The chances of sharing or the recombination is less. So अगर ये branch point इस तरफ जाएगा, ये branch point इस तरफ जाएगा, so the amount of recombinant formed will be more. है ना? जो sharing होगा वो आपका ज्यादा होगा. Is it clear?
Yes, ma'am.
Okay, yeah. So what I'm saying is that this branch migration means this holiday junction will move this side and this one will move this side, so that you can have maximum of sharing between these and these. Okay, now next. So what is happening now? The RUV A and RUV B. So RUV A का काम होता है to recognize the holiday junctions. Okay, now this is how your holiday junctions looks like. You can see that there are 4 different DNA strands are there: green, red, yellow, and blue. Okay, so this is how your holiday junctions look like. So RUV A क्या करेगा? It will, wherever it sees that holiday junction is there, it is going to bind over there. It's a tetramer. Okay, then B, it is actually a hexamer. Okay, now RUV B is having the ATPase activity. So ये क्या करेगा? It will keep on moving. Okay, it will keep on moving so that the DNA will get a space to cut. Now, can you see this is the crystallography structure? Not that clear, but yeah, you can still see that here blue and pink is there, green and red is there. So again, here 4 are there, and this is a tetramer RUV A protein which is binding at the holiday junction. Now, this is the same thing which is depicted in the ribbon model. Is it clear, guys? Can you see how RUV A has, has bind over here at the HJ holiday junction, and 2 in between RUV B has bound at the sidewise. Now, the question should be asked is that the RUV B can bind over here also, right? Is it possible? What do you think, guys? Is it possible for RUV B to bind in this direction also? Yes, it is possible. If you remember in the holiday junction, either we are going to have the cut like this or cut like this. Okay. So it depends on that, like how this helicase is going to move. Okay. Okay. Again, this is another figure, like for the better understanding. So this is how in the electron micrograph you see, and then later we can just expand the image and you can put the color so that it becomes easy to understand. Okay, this is the atom model. Again, you can see the 4 strands of the DNA. This is your tetramer, and then sideways you have got the hexameric ring of the group B. Okay. Now, so group B has got the ATPase activity. Okay, so it can bind in, it will bind in those such a way that where it has got the low affinity for the DNA. Are you getting? So group B is an ATPase active in the presence of DNA compared to group B, and it has got low affinity for the DNA. So they are the proteins which thought to form the hexameric rings on the exit points of the newly formed DNA duplex. तो जैसे ही जहां पे खत्म हो रहा है आपका हॉलिडे जंक्शन, वहां पे आपका ग्रुप बी आएगा, और उनका काम क्या है? डीएनए को खींच के निकालना फ्रॉम दी योर हॉलिडे जंक्शंस. Okay, able to follow, guys? nahi? Or you can just understand in such a way that since it has got the ATPase activity, so जैसे जैसे वो ATPase activity रहेगा वैसे वैसे ब्रांच आपका इस तरफ मूव होगा. Okay, so just understand in such a way that अगर आपका holiday junction जो है it has to move this side as part of the branch migration, then group B will bind to the other side. ये push करने का काम करते हैं to the holiday junction. Are you getting the point? So this two will be pushing in this direction and these two will be pushing in this direction. So उससे क्या होगा? आपका holiday junction finally यहाँ पहुंच जाएगा, इसका यहाँ पहुंच जाएगा. Yes, able to imagine? You can see.
Yes, ma'am.
So it has got the ATP. So ATP hydrolysis होगा. It is pushing your group B to push the entire group A and these two so that it goes in this direction. Sorry. Okay, it goes in this direction. So this will go in this direction. Okay. So eventually what will happen? Finally, wherever the possibility is there, it is going to form the holiday junction. And we know that in the case of holiday junction, first the vertical, it is going to give you the crossover products. Horizontal means it is going to give you the patch products or the non-crossover products. Now keep this thing in the mind, okay? Keep this thing in the mind. क्योंकि ये करने कौन वाला है? Group C, which is also known as your resolvase. Okay, this is also known as resolvase. The thing is that since here we have got the two holiday junction, the possibility of forming the recombinants is so many are there. Okay? Can anyone tell me what are the possibilities? Yes? कोई try करना चाहेगा? Tanvi, would you like to try? I'll tell you 1 और 2 का मतलब क्या होता है. So 1 means what? वो यहाँ पे add होगा और ऐसा कट देगा. अगर 2 में add हुआ तो horizontal कट देगा. ठीक है? So 1 means giving you the vertical cut of the DNA. Horizontal means that is your 2. So horizontal. Now, would you like to try what are the different combination possible over here? Tanvi, would you like to try? Akshay, you?
Okay, ma'am.
Just try it. Let's see whether you're getting the answer or not. कुछ नहीं होता अगर गलत भी होता है तो. हाँ.
So for the first one, for the left one or the right one, ma'am?
See, I'm talking about the possibilities, right? So you need to consider both at the same time.
Okay.
हाँ, because inside the cell it is not like ये पहले होगा and ये पहले होगा. जो भी होगा दोनों साथ में होगा. हाँ. So what are the different combinations which is possible?
So how should I tell, like with the color or like how, ma'am?
With the number. Color से तो मैं confuse हो जाती हूँ.
Ma'am, but there are 4 strands, right?
There are
4 strands.
so in the holiday you are going to have 4 strands only, right?
Should we come?
Over here also 4 strands are there. Black, gray, pink and red.
Yes, ma'am. But in the picture there are only 2 numbers, like 1 and 2. So that's why.
Now 1 means it is a vertical cut, 2 means horizontal cut. Okay, I'll give you one hint. You can have combination like okay. So these are the 4 scenarios. I'm just repeating again. These are the 4 scenarios where we can have 1 having both vertical cuts, 2 having both horizontal cut, right? Then third scenario where 1 is vertical and horizontal, horizontal and vertical. So now what do we have to see is that among these 4, which is going to be your splice recombinants and which is going to be your patch recombinants. Okay, clear? Now,
Yes.
look, let's see what are those scenarios. So first one, what is the first scenario? 1, 1, correct? So let's see in the case of 1, 1, what is happening. So your RUV C is coming and binding like this. Can you see? Can you imagine how it is coming and binding? Okay. Now, imagine that holiday junction, it is a vertical cut. Okay, it is a vertical cut. Then how it is going to resolve? Resolvation means it is going to make sure that after the cut, the DNA is also ligated, if you remember, right? So can you see? It is going to bring it like this. So we have made the vertical cut over here. Now, when it is resolving the DNA, now can you see what is happening? It has pinched all the 4 and ligated. Okay. During this ligation, what had happened is that the gray and the other strand is your normal. Can you see or understand? Okay, we'll see all and then you tell me whether you are able to follow or not. Okay. Now, in this case, the RUV C is in this direction. Okay. Is it in this direction? Now, just see what it has done. It has made the cut and now it has joined over here itself. Oh, sorry. Now over here. So this is both where 2 is happening. Okay. So RUV C is there. It is making a cut over there and it is joining like this. Okay. Clear? जो आपका स्ट्रैंड इन्वेड किया था इनके बीच में, राइट, उसको कट करके वहां छोड़ दिया और यहाँ पे ज्वाइन हो गया आपका. ओके, सो इन दिस केस योर फर्स्ट ऊपर का स्ट्रैंड एंड नीचे का स्ट्रैंड वाज नॉट इन्वॉल्व्ड. करेक्ट?
Yes, ma'am.
ओके. थर्ड सिनेरियो वेयर यहाँ पे आप देख सकते हो, यहाँ पे आपका सेकंड हो रहा है, सॉरी यहाँ पे शायद 1 होगा. यस, यहाँ पे 1 हो रहा है. ओके, दे हैव हाइलाइटेड, राइट? सो हियर इट इज 1 एंड हियर इट इज योर सेकंड. 1-2 सिनेरियो ओवर हियर इट इज, राइट? यहाँ पे 1 है, यहाँ पे 2 होगा. ओके, सो अगेन लुक इट ओवर हियर. तो सेकंड में आपका नॉर्मली रिजॉल्व हो गया है एंड फर्स्ट में ऐसे हुआ है. ओके, so because of which what had happened, cut and they have gone under the crossover. Can you see? If you have understood the holiday junction, तो ये clear हो जाएगा. You have to imagine that this strand is getting opened and it is coming in that particular figure like what we have discussed. ऐसे वाला. Okay, ऐसे वाले में you have to imagine this. एक ये स्ट्रैंड है आपके पास, एक ये हो गया, एक ये हो गया. You need to imagine in that way. Then if you see the crossover is happening over here and finally the strand, this is going to be different and this is going to be different. Okay, so here you can see that crossover products are present. ये देखो, ये वाला आपका इसके साथ है combined, this one with this, this one with this, this one with this, and this one with this. So all the 4 strands are getting changed. Yes?
Yes, ma'am.
Okay. Next, यहाँ पे second and यहाँ पे 1. This is last scenario, 2, 1 ka scenario. So again, so here it will be normal fusion, but again here everything is getting fused to each other. This one with this, correct? This one with this one with this, and this one with this. So again, everything is getting cross. So here again, we are going to get a splice recombination products. Clear? Okay, so that's all. So now it is clear, like how are we going to get all those 1, 1, 2, 2, 1, 2 and 2, 1 ka crossover products and non-crossover products? Is it clear? So at the end, what is happening? What is the takeaway message? That at the end, if there was a double strand break, okay, in case of prokaryotes, with the help of these proteins. So how many proteins are involved in this? So we talk about rec A, B, C, D, and then we talked about room A, B, and C. Correct? So in your exam, if it's a theory exam, then you need to talk about all this rec A, B, C, D, room A, B, C, and of course, obviously about the holiday junctions. But in order to understand the crossover products, this combination you should remember. Of course, diagram banane ke liye koi nahi puchega. Just remember that this is how it has to be done. Clear? I'll show, like maybe in the next lecture, I'll show how to answer these questions, because there are multiple choice questions based on these topics. So you'll get an idea once I share those questions with you. Clear? And the PDF which I have shared with you about the DNA replication wala, in that everything is given. Okay, ye bhi questions hain usme. So just go through once and check yourself whether you're able to answer those or not. Okay. So thank you guys.